Okay, welcome to the third lecture in the Conservation Genetics series. In this one, we're going to take a look at changes in population genetic diversity. In the previous podcast, we've taken a look at populations and what genetic diversity is, and genetic diversity in populations, and we saw how the Hardy-Weinberg principle suggested that populations' genetic diversity should remain constant given certain conditions. I also said that it's very seldom that you would actually get those conditions to satisfy Hardy-Weinberg conditions. So there is almost inevitably some changes in the genetic diversity or genetic composition of populations as time goes on. And some of the processes which change the genetic composition and diversity of populations are selection, random genetic drift is another one, migration or gene flow between populations is another one, and finally mutational disequilibrium. So I'm going to go through these different evolutionary forces which will disrupt Hardy-Weinberg equilibrium and change the genetic diversity and composition of populations. So basically change the allelic frequencies which exists in the populations. So there are two main evolutionary forces which cause changes in allelic composition and therefore genetic composition and diversity above all the other forces, and that is selection and random genetic drift. So first of all, selection. Selection arises due to the differential ability of genotypes / individuals within a population to survive and leave offspring. And this differential ability is based on the interaction between the phenotype or the characteristics of that organism, the interaction between the characteristics of that organism and the environment. If the environment is conducive to that particular organism and its genotype, then it will leave more offspring than an organism whose genotype is not so much in sync with the environment. So selection results from a reproductive advantage, and most important, it is not random. So certain genes in different individuals within the population, if those genes are the same, then those individuals are going to be favored by the environment. In effect, they're going to be selected by that environment. So if you have an example, for instance, of a small mammal like a rabbit in a environment, a snowy environment where everything is white, the rabbits which are white in color or have white fur will tend to survive better because they are more camouflage, and because they survive better, they will leave more offspring and therefore contribute more to the next generation. So their offspring will be carrying the genotypes and the alleles for white coat color, and the individuals which have the alleles for brown coat cover will stick out in the snow and will be predated upon, and they will not leave offspring. So selection will choose for specific alleles, and it is not random, so those alleles will be represented more in the next generation. Okay, so selection. So the rate of evolutionary change, or the change in a population over time due to selection, is very much in proportion to the amount of genetic diversity available. So if you have a large amount of genetic diversity and you have individuals which may be favored by the environment, then those individuals are going to increasingly represent their alleles in the next generation. When you have low genetic diversity, there may be less individuals. Some individuals may not be able to take advantage of the environment, and so quite often no one individual can survive, or no one individual can monopolize the resources under that environment. So there is no real selection pressure going on, changing the allelic diversity and the genetic makeup of the population. So the rate at which selection works on a population is directly proportional to the amount of genetic diversity within that population. If there's less genetic diversity, there is less for the selection to work with, and therefore there is going to be a lower rate of change or evolution or ability to change in the face of changing environmental pressures. So selection forces will increase the variation between populations. The reason why selection increases the variation between populations is that, for instance, population B down here will have very different environmental factors to population A up here. Well, it will have different environmental factors. So A may have it's further away from the equator, so it may be drier. B may be wetter. And so the wolves in population B, because you remember that this is a map of the populations of the Ethiopian wolf, the wolves in population B may have genetic makeup which allows them to survive in a wetter environment compared to the wolves in population A, for example. And selection is the process which selects for alleles which allow wolves to do better in a wetter environment. On the other hand, migration, which is another evolutionary process, this actually decreases the variation between populations. So if our population, say these adjacent populations, had individuals migrating between them, then the genetic makeup within those populations is going to be more similar than if they were isolated and there was no migration. So populations between B and A are much more likely to be different than populations which are closer together, because migration is much more unlikely between B and A than it is from the closer populations. So selection increases the difference between population. It increases the alleles increases the difference in the alleles which are found in population B compared to population A. But migration will act to even those differences once again. So evolutionary changes can take place over time, and if the change in the environment is slow enough, then natural selection can actually keep pace with those changes in environment. So you may have a difference in environment after 100 years, which if you went back to time zero, no individual within the population could have survived the different population you have after 100 or 1,000 years. So what happens there is selection works to change the population to allow it to survive the changing environmental conditions over time. And this is known as adaptive evolution. And there are examples of this in the fossil record that we can see. And it makes you realize that the changes in the global climate, which humans are instigating, can have a big effect on genetic diversity in populations, because it is happening so rapidly. It's not allowing the time for natural selection to work, and therefore adaptive evolution to work to allow those species to change to those different environmental characteristics around the world. Okay? But this is a term which you should know, adaptive evolution, and it basically states that selection through evolutionary changes allows individuals to cope with changes in conditions, environmental conditions, gradually over time. All right, so we've seen that high genetic variation or great genetic variation within populations is very important in allowing that population to adapt to changes. This can also be termed as heterozygosity or vigor within the population. If a population is said to have high vigor, it's quite often because it has high genetic variation or high heterozygosity. Okay? All right. Now, another point which we need to make with selection is that it works best in populations which have high genetic diversity, high amounts of allelic diversity. Now, the populations which tend to have high levels of allelic diversity tend to be the larger populations, because the larger the population, the more individuals there are, and therefore the more room there is for different alleles to exist. So selection tends to work best in larger populations. So selection tends to be the key evolutionary process in larger populations of plants and animals. When we move to smaller populations, on the other hand, the major evolutionary force causing change in allelic diversity tends to be random genetic drift. And I'm going to tell you why first of all, I'm going to tell you what random genetic drift is, and then I'm going to tell you why the major evolutionary force, it is the major evolutionary force in small populations rather than selection. All right, small populations. So we would have small populations which have small numbers of individuals within them for different reasons. Two of the main reasons are unfavorable environments result in a rapid reduction in population size. This is known as a bottleneck, and populations which have reduced due to this change in unfavorable environments being imposed upon them. This is known as the bottleneck effect. Populations which are new and inhabit a new place are quite often founded by small numbers of individuals. So their founding populations are quite often very small. So populations may be small because they are new, and this is known as the founder effect. So small populations are created by these two main processes. First of all, a reduction in a previously large population size, known as the bottleneck effect, and populations which disperse from the main population or are separated from the main population, a small number of individuals, and this is known as the founder effect. Both of these populations, no matter how they arise, are small. So they both suffer from processes which occur in small populations, and that process, that dominant evolutionary process, is the random genetic drift. Okay, so just explaining the bottleneck effect more, a small surviving population is unlikely to represent the genetic makeup of the original population. So when you eliminate large numbers of individuals from a population, you are going to cut down on the genetic makeup of that population. So much so that it's been estimated that if you reduce the population by 90%, so there's only 10% of the individuals left, you would quite likely only have 50% of the genetic diversity or the allelic diversity left within that population. And the reason for that is that some of the alleles are rare. They only occur in a few individuals within the population, and because of that, there is little chance, or there is a lower chance, that the individual which survives the massacre with the change of the environment carries those rare alleles. So large numbers of the less common alleles will be lost from the population due to a rapid reduction in population size. And that rapid reduction could be due to habitat loss, overhunting, etc., etc. So an example of the bottleneck effect is the South African cheetah. It's considered to have lost its genetic diversity due to multiple bottlenecks. I'm not too sure how those bottlenecks occur, maybe through changes of environment, dry periods or warm periods, or reduction in population sizes due to persecution by people. So it basically has a very low heterozygosity, a very low allelic diversity within its population. The effects of this can immediately be seen. Cheetahs generally, these South African cheetahs generally tend to have very poor sperm count, up to 10 times lower than related cats. 70% of those sperm are abnormal, and they find it very difficult to perform their function. So therefore you would have very poor breeding efficiency. You would tend to have high infant mortality when the sperm, which are deformed and abnormal, either cannot make it to the egg, or if they do make it to the egg, they have deleterious recessives, which means the proteins which are necessary to create the kittens are not there, or they are defective, and so the fetus will abort. Okay? It also means that cheetahs are very susceptible to diseases. So if there is a disease which infects a cheetah population, every single individual within the population is usually susceptible to that disease. And that can be very problematic. If a disease gets into the population, it can wipe out a population. So South African cheetahs are a good example of very low heterozygosity within their populations due to a bottleneck effect, and the effects can readily be seen in the cheetah populations. All right, moving on to the founder effect. The founder effect is where small numbers of individuals break away from the main population and they colonize a new habitat. And the example that we have there is a European or Mediterranean species. I'm not too sure what it is, but it's colonized the Mediterranean regions of South America and North America. And if you look at the numbers in the balloons here, there are 80 inversions, or 80. An inversion is a type of noticeable genetic mutation. There are 80 of them in the main European populations, but there are only 20 in the South American and North American populations. So that indicates that not all of these genetic features have made it to these other populations, and that is probably due to the fact that it was a small number of individuals, the total of which only contained a quarter of the allelic diversity within the European population, and carried it to the new populations. So this is an example of a small population having reduced genetic diversity due to the founder effect. Another example of the founder effect is in Martha's Vineyard off the coast of Massachusetts on the eastern side of the United States. Martha's Vineyard was a small island which was only settled by a small handful of English families, only a few people. And as a result, the recessive deleterious alleles which control for deafness was overrepresented in that population, and they expressed, and it meant that the population had a rate of deafness of up to 30 times more than that of the mainland population in the United States. So the founder effect there meant that genetic diversity was so low in Martha's Vineyard that deafness, which was the result of a deleterious recessive pair of alleles, meant that it was much higher in Martha's Vineyard compared to the mainland. Okay, I'm not going to go into the example of the Galapagos finch. All right, so small populations are formed either by the bottleneck or by founder effects, and they tend to have fairly low numbers of individuals, and quite often they have moderate to small amounts of genetic variation because they are such small populations. Now, they become subject to random genetic drift simply because there are small numbers of individuals in the population. So what is random genetic drift? It's basically an arbitrary fluctuation of allelic frequencies due to errors in sampling of gametes that form the next generation, results in some of the rare alleles not being passed on to the next generation and therefore being eliminated from the population. So how does that happen? It happens through sampling areas. With a big population, you would get pretty much an equal distribution of an allele which has a 50% chance of being passed on to the next generation. So for example, when you are tossing a coin and you toss enough coins, you would maybe get 500 heads, 500 tails. And the heads and tails are going to be represented equally in the next generation or in the result. But if you only throw 10 times, you are going to get biased numbers of heads and tails, because it's very unlikely for you to get 5 heads and 5 tails exactly. You would more likely get maybe 7 heads or maybe even 8 heads and 2 tails, or the other way around, 8 tails and 2 heads, or maybe 3 and 7 or 4 and 6. Okay, so you would much more likely to get a biased passing on the alleles from one generation to the next in a small population. In a large population, there are enough individuals, each passing on their genes, that the rare alleles have a good chance of being passed on. But if you have a small population, the rare alleles have a much smaller chance of being passed on. So this unequal distribution is referred to as sampling errors. And what it means is that in small populations, allelic frequency will fluctuate, and in some cases, if it fluctuates to such an extent that it goes below 0, that means it has disappeared from the population. So that allele no longer occurs in the population, so it can no longer be passed on. So it is lost. And if you have a locus which has the alleles big A and small a, then it no longer has the allele of small a. It only has the allele of big A, because big A is the only allele which is left in the population. So as a result, we can say that locus now has lost its heteromorphic status, and it is now monomorphic, and that locus has become fixed to this one allele. Okay? So this occurs because of the small population size, meaning that as the frequency of the allele gets smaller and it becomes rarer, there is less chance of it being passed on to the next generation. And because it becomes less and less likely to be passed on to the next generation, eventually it just disappears altogether. So big A, this allele big A, looks like it's going down as well. And eventually there will be a point where, if there's only 10 individuals in the population, there will be a point that only small b allele is actually passed on to the next generation, and big B allele falls to 0, and it is no longer represented in the population. So the locus or the gene has been fixed on small b allele. Okay? So this is known as random genetic drift. Now, this, unlike selection, is not controlled by the environment. It is an entirely random process. So a gene may be just as beneficial to the organism in the current environment as another gene, but it can be lost anyway. And it can be lost anyway because it's an entirely random process. So once an allele by chance gets undersampled, say it only gets picked 3 times out of 10, okay, or maybe 13 times out of 50, then suddenly it's in danger of being lost, because its chances of being selected again in the next generation now becomes smaller. All right, so through that process of random genetic drift, genetic variability is depleted in small population due to random loss of alleles. So more and more genes become fixed with one allele, and heterozygosity of alleles within the genome becomes smaller and smaller. So genetic variability becomes smaller and smaller. Because genetic variability becomes smaller and smaller, the population will become less fit. It will not be as reproductively successful. So in the short term, like the cheetahs, deleterious alleles become more and more expressed. And so reproduction becomes more problematic as, say, sperm rates begin to decline and there is more abnormal sperm, or the eggs become less viable, or there's less viable eggs. So reproduction becomes more and more of a problem. And as reproduction becomes more and more of a problem, the population size will decrease. As the population size decreases, more alleles are lost, okay, through random genetic drift. So, in summary then, loss of genetic diversity, which results from the loss of numbers of individuals, causes random fixation of alleles, and many of those alleles become fixed in the population because the other allele at the same gene is lost through random chance. Okay? And many of those alleles in the population may be deleterious, which reduces the vitality of the population. Okay? So the deleterious alleles become more expressed in that population and leading to problems in the vitality of the population. Another effect of random genetic drift is the rapid differentiation between populations. So because an allele is lost in these small populations due to random genetic drift, the same random chance won't happen in a separate population, and instead another allele or a different allele will be lost. So the genetic variation or the genetic composition between populations tends to decrease rapidly. Okay? So their comparison or the common alleles between the populations begins to fall. So you get less alleles in common between the populations, and so those populations can be said to be diverging. So small populations will tend to change faster. Okay? And we can work out this loss of genetic diversity over time by looking at the effective population size. Now, the effective population size denoted by NE, we're going to talk about in the next podcast, but suffice it to say that it is the population, or the number of individuals within the population which are actively reproducing. And because the population at any one time, it may have older individuals which are no longer breeding, or younger individuals which have not yet begun to breed, the effective population size is always going to be less than the actual or census population size. So it's the effective population size that we really look at when we're looking at whether a population is small or not. And this graph shows that if your effective population size, or NE, is very small, then the amount of genetic variation or heterozygosity which remains after a number of generations falls very rapidly. But if your effective population size is large, then your effective or your heterozygosity will remain high over these generations. Okay? So with a small population, you will get a slight loss in genetic variability over time because of that random genetic drift. But this slight decline over the generations will quite often be compensated for by mutation rates and so on, so that genetic variability will remain high as long as the decline is not very pronounced. Okay? So it's not just in one season or 1 year that we need to look at that effective population size, because apparently effective population size will vary from year to year. So if we take the example of a population of annual flowers, in 1 year the rains may not come, and so the number of individuals which survived to set seed and produce that seed may have only been 10 within that population, and that in another year the rains were very good, and so 10,000 individuals survived to disperse their seeds to form the next generation. So you may have a population which effectively fluctuates from 10 to 10,000 and other numbers in between depending on the season. So if we look at 4 generations, or the number of look at how the genetics in the population fluctuates over time, we may find that the population loses about 5.5% of its heterozygosity, because if we can calculate the amount of genetic variation remaining after 4 years by summing up all the changes in genetic effective population size, and it gives you 0.945 of the genetic diversity, of the total genetic diversity, actually makes it through these 4 generations. Okay? But the loss of the genetic diversity is greatest when the effective population size is smallest. Okay, so you can see 1 over 20 is going to be a much larger figure than 1 over 20,000. So overall, the population losses of its heterozygosity of this organism over 4 years or 4 generations was 5.5%. But the majority of that loss of heterozygosity occurred in this year or this generation when the population size was only equivalent to 10. Okay? So, loss of genetic diversity is particularly pronounced when your population size fluctuates over time. If your population size remains constant and high, then the genetic diversity is not going to be lost. But if your population size reduces to a low number and then rebounds to a high number, or reduces to a low number again, then it's going to lose more genetic diversity over time, because those small population sizes represent times when less genetic diversity is available to be passed on to the next generation. Okay? All right, we also need to mention that random genetic drift and loss of genetic diversity is different when you have different forms of genetic material. So if you have a diploid genetic material, you would have a particular rate of genetic loss, which is 1 over 2 times the effective population size. If you have a tetraploid, then it's 1 over 4 times the effective population size. So in other words, the rate of genetic loss of genetic diversity is much lower. And if you have a haploid, the rate of genetic loss of genetic diversity through random genetic drift over time is much higher. It's 1 over the effective population size. All right, so the different forms of the genome of the organism also has a big impact on the rate of loss of genetic diversity. So diploids and tetraploids will maintain their genetic diversity longer than haploids and other organisms which have different forms of genomes. All right, random genetic drift can also be balanced off by selection. Now, in small populations with small variation, selection is less of a evolutionary force, but there can be some balancing off through selection. So these are the different ways in which selection can balance or retard or stop the loss of genetic diversity by heterozygote advantage, when particular heterozygotes have a higher fitness than either of the homozygotes. So if you have an organism which has the 2 different alleles at the particular locus, it's a heterozygote in a diploid organism. If it has a higher fitness in that environment, then it's going to maintain itself better in the environment compared to the homozygotes. Okay? So if that advantage is pronounced, then heterozygotes are going to remain longer. This is, we can also call this hybrid vigor. Okay. Frequency-dependent selection. When a genotype is favored at one frequency but disadvantaged at another. Okay, so if a genotype, when it is very common, it tends to be more preyed upon, or it tends to be, the population of that genotype tends to be reduced, but the other genotype, which is at a lower frequency, is preyed upon less. That can balance out the imbalance in genotypes. So the alleles which are more common in the population are then reduced, and that allows the alleles which are less common to be able, more of a chance to be transferred into the next generation. Okay, so it's like density-dependent mortality. If you have more organisms, more individuals of a certain organism, it is more likely to be preyed upon, giving the less competitive individuals, less competitive species, more of a chance to survive. Okay. You can also have selection in different environments. So if a population occurs in a seasonal environment, one genotype may be very good in one season, and the other genotype may survive better in the other season. Okay, so different genotypes may have different successes in breeding in the different environments. So in that way, you would have almost different environments in the same population, which will allow one genotype to dominate one part of the year, the other genotype to dominate the other type of year. So over the course of the whole year, both genotypes can survive with one another. And finally, we would have associative dominance, where even though there's blocks of loci containing deleterious alleles, the effects of those loci with deleterious alleles is masked because those blocks are associated with other alleles which are not deleterious. So the loci will still act as if it is heterozygous. That's because the loci are grouped together and their effect is masked by another associated loci. Okay, so this can all retard the loss of genetic diversity and the effects of low genetic variation. So which is the major evolutionary force? As I've said, selection is the major evolutionary force when population sizes are large. Okay, so for example, if the population size is greater than 10 in this case, then selection is the major evolutionary force. If it is less than 0.01, then random genetic drift is the major evolutionary force. If it's between 10 and 0.01, both random genetic drift and selection are a major evolutionary force in that population, maybe at different times, in different generations. Okay? So in summary for this particular little video on conservation genetics, there are four main forces of change in genetic diversity: selection, genetic drift, migration, and mutation. Selection is important in large populations, and it acts to change the genetic variability and the genetic composition. Quite often the genetic variability is maintained because there is a large enough population so that sampling error is not an issue. Genetic drift is important in small population. Genetic drift is the loss of genetic variability through the loss of particular alleles due to sampling error, creating a situation where allelic frequency falls to such a point that it has an extremely low chance of being represented in the next population. So genetic drift is important in small populations, and small populations can come about through either bottleneck and a founder effect. Population size and therefore genetic variation, because the larger the population size, the more the genetic variation, becomes reduced in populations by the bottleneck effect or the founder effect. Okay, so thank you very much for listening, and good luck with the understanding. Bye.